In the past few years, gene therapies have taken off. They represent a major shift in medicine. Instead of just treating symptoms like many traditional drugs, gene therapies aim to correct the underlying genetic cause of a disease by modifying a person's DNA to replace missing or malfunctioning genes. Its most recent success is with Leber congenital amaurosis, or LCA, the first genetic disease to be treated with an FDA-approved gene therapy. However, leading up to this breakthrough success was decades of research, from basic science to translational studies to clinical trials. LCA is a retinal degenerative disorder characterized by severe loss of vision at birth. It is inherited in an autosomal recessive manner and affects 2 to 3 per 100,000 births, making it one of the most common causes of inherited blindness in children. One of the most common causes of this disorder is a mutation in the RPE65 gene, which was first described in 1997. RPE65 is a 65 kilodalton protein that's expressed in the retinal pigment epithelium, the RPE. Now, they were fairly confident that this was a protein that was responsible for LCA, but the function of this protein was still unclear. A study conducted by Redmond and colleagues investigated this by knocking out the RPE65 gene in mice, which resulted in massive changes to the rod photoreceptors. But first, let's backtrack and talk about the visual phototransduction pathway. In the back of the eye, right inside the RPE, lies a layer of rod photoreceptors. Inside these folded membranes are pigments, which we call rhodopsin. Rhodopsin is made of two components. This here is retinol. It's one of many forms of vitamin A and is a polyene chromophore, which means it has an absorption spectrum in the visible region. The retinol is bound to a protein called RGR-opsin, a retinal G-protein coupled receptor. The absorption of a photon results in a photoisomerization of the retinal chromophore from 11-cis-retinol to all-trans-retinol. This structural change then causes a change in the conformation of the opsin G-protein coupled receptor, which eventually activates the associated G-protein, known as transducin. Transducin can then go on to activate the rest of the visual transduction pathway. Now, following the activation, the rhodopsin is inactivated by the hydrolysis of all-trans-retinol from the opsin. Attachment of a fresh molecule of 11-cis-retinol derived from vitamin A or from all-trans-retinol regenerates the rhodopsin, resetting the system so it's ready to absorb another photon in what is known as a bleach and recycle process. Now we can go back to the knockout mice. They found that, without the RPE65 gene, we get an accumulation of the all-trans-retinol esters and a lack of the 11-cis-retinol, indicating that RPE65 may be the effector enzyme in the key isomerization step needed to reset the visual system. Later, the Ma group at the University of Oklahoma determined that RPE65 in fact was the enzyme that catalyzed the conversion from all-trans retinol ester to 11-cis-retinol, which eventually gets oxidized into 11-cis-retinol. Armed with this basic biochemical knowledge, Penn researchers Dr. Jean Bennett, Dr. Catherine High, and Dr. Albert McGuire, along with many other collaborators, began testing a gene therapy approach, which is called voretigene neparvovec, or LUXTURNA. Now, how does this work? Voretigene neparvovec is an AAV2 vector, an adeno-associated virus. It carries its genome plus the human RPE65 cDNA sequence and gene expression drivers into the host cell, but does not integrate into the genome, instead persisting as an extra chromosomal episome. The viral DNA is then translated into protein, providing a functional copy of the RPE65 protein to replace the defective one. So they purify the virus and then they inject it, 1.5 times 10 to the 11th vector genomes in 0.3 milliliters through a subretinal injection in each eye. After 1 year of their phase 3 clinical trial, they measured the change in performance of 29 individuals using MLMT testing, an obstacle course which assesses the ability of a patient to navigate at different levels of environmental illumination. A median Lux score change of 2 was observed in the LUXTURNA treated group, while a median Lux score change of 0 was observed in the control group, with no serious adverse effects. Now, after over 25 years of study and the work of many researchers at Spark Therapeutics and the Children's Hospital of Philadelphia, we have LUXTURNA. While this is not yet a complete cure, and there's still work to do in decreasing the risk of eye infections, retinal tears, and cataract development, as well as lowering the heavy price tag to improve patient access, this breakthrough gives hope to those with LCA and further opens the door to the potential of gene therapies.